As part of the program of the bacteriology class, and as members of team number 1, we're going to talk about the test-loop-mediated isothermal amplification coupled with nanoparticle-based lateral flow biosensor assay for detection of Haemophilus influenzae B. Haemophilus influenzae is a gram-negative small 0.3 micrometer to 1 micrometer Coccobacillibacterium from the Pasteurolaceae family. It is also facultatively anaerobic, pleomorphic, campnophilic, and non-motile. Bacteria of the Haemophilus influenzae species are broadly classified into encapsulated and non-encapsulated types. Its natural history is divided into several stages, beginning with exposure, in which human beings are the only host for this bacterium, and the principal mode of transmission of Haemophilus influenzae is through inhalation of respiratory secretion droplets from infected individuals or by direct close contact. The molecular changes are adherence of Haemophilus influenzae to host cells by protein H, and Haemophilus sulfate fibrils, and IgA proteases that destroy IgA in mucous membranes. The biochemical, physiological, and anatomical changes include the production of pro-inflammatory cytokines and alteration of tumor necrosis factor alpha signaling that causes an unregulated inflammatory response that presents as cardinal points of inflammation, pain, and fever. The only sign is high-grade fever, and for the most common symptoms, we may find chills, purulent cough, shortness of breath, chest pain, lethargy, generalized body aches, headache, altered consciousness, photophobia, nausea, vomiting, stiff neck, respiratory distress, and sore throat. As for the children, they may have non-specific symptoms such as irritability, drowsiness, and poor feeding. The Haemophilus influenzae type B disease causes generalized infections in the respiratory tract that without treatment can develop into meningitis, pneumonia, and bacteremia. After the disease, hopefully the subject will have a healthy recovery, but prior to the development and use of Haemophilus influenzae type B conjugate vaccines, 20 to 30% of survivors had some form of permanent sequel, ranging from mild hearing loss to intellectual disability.
The main variance factor involved in the pathology caused by Haemophilus influenzae type B is its polyribosyl ribitol phosphate capsule, which makes it resistant to phagocytosis by polymorphonuclear leukocytes, in the absence of a specific anticapsular antibody, and triggers bacterial proliferation. As mentioned before, it has proteins such as protein H and Haemophilus surface fibrils, which are adherence factors that allows them to adhere to the host cell and proliferate. Other variance factors of Haemophilus influenzae are pili, lipoligosaccharides, quantum sensing system, and a two-component signaling system which promote adhesion to the host cells and biofilm production, highly related to its antibiotic resistance. It also has IgA proteases, which destroy the IgA that protects mucosal barriers in the nasopharynx and stimulates production of a pro-inflammatory cytokine, and alter tumor necrosis factor alpha signaling. So now, about the test. This test detects a DNA sequence of a gene encoding for an OMPP6 of Haemophilus influenzae. OMPP6 is a highly conserved gene in the bacterium. It is a suitable target for Haemophilus influenzae identification than other genes. In fact, it is a gene encoding for an outer membrane protein P6, which are outer membrane surface structures and important in colonization of the human host and in pathogenesis of infection. P6 is exposed to the surface of the bacterium. The protein expresses epitopes, which are present in all strains of Haemophilus influenzae, both typable and non-typable on the surface of the intact bacterium. LAMP tests employ a DNA polymerase and 4 primers that recognize 6 distinct sequences on the target DNA. An inner primer containing sequences of the sense and antisense strands of the target DNA initiates LAMP. Strand displacement DNA synthesis primed by an outer primer releases a single-stranded DNA, which serves as a template for DNA synthesis, which produces a stem loop DNA structure by the hybridization of outer and inner primers. Then, displacement DNA synthesis is initiated by cycling and hybridization of one primer to the loop, obtaining as products the original stem loop DNA and a twice as long new stem loop DNA. This cycling reaction continues until accumulation of 109 copies of target DNA. The final products are stem loop DNAs with several inverted repeats of the target and cauliflower-like structures with multiple loops. Because LAMP recognizes the target by 6 distinct sequences initially and by 4 distinct sequences afterwards, it is expected to amplify the target sequence with a high selectivity.
A standard lateral flow strip LFS consists in 4 main sections: a sample pad made of cellulose, conjugate pad made of glass fiber impregnated with a lava particle and a receptor for the analyte, detection pad, a nitrocellulose sheet where test line TL and control line CL are printed, and absorption pad also made of cellulose. The use of crimson red dye streptavidin-coated polymer nanoparticles allow optical detection of the test result. In the assay with LFS, the sample is added on the sample pad, where the liquid will start flowing to the conjugate pad, where the analyte, if present on the sample, will be linked to the transducers, previously conjugated with a bioreceptor specific to the analyte. The conjugate, rehydrated by the liquid, will flow by capillarity across the detection pad to the absorbent pad, passing through the test line, where it will be captured only if the conjugate has the analyte attached, and to the control line, being always captured to evidence that the assay works. The whole experimental steps of LAMP-LFB can be completed in 52 minutes, which include 20 minutes for genomic template preparation, LAMP reaction time of 30 minutes, and LFB analysis of 2 minutes. LAMP is done at 65 degrees Celsius that stays constant throughout the whole experiment. It's very common that the sample to be used is sputum, commonly the deep cough. Sample of the early morning is collected before eating or drinking anything to avoid bias in interpreting the results. At first, the patient needs to rinse out the mouth with clear water of 10 to 15 seconds to eliminate any contaminants in the oral cavity. After expelling saliva, the patient then breathes in deeply 3 times to cough at 2 minutes intervals until bringing up some sputum. The sputum is then released in a sterile well-closed container provided by the medical professional to the patient. In some settings, the procedure can be repeated until 10 to 20 milliliters of sputum sample has been collected. Routine sputum culture requires that one sample is collected and sent to the lab on the same day of collection. If the patient leaves the specimen in the refrigerator after collection, there is often a tolerance range, which may run well over 24 hours.
The DNA needed from sputum samples can be obtained by both dititritol and enzymatic treatments. The reactive sulfhydryl groups of DTT have a mucolytic function and are used to reduce mucoprotein disulfide bonds. DTT is commonly used in standard microbiology to homogenize sputum samples, enhancing the isolation of microorganisms, and has been also adopted in different studies for microbiota analysis. Controls. For the purpose of assessing the effectiveness of Haemophilus influenza LAMP primers, the LAMP reactions can be conducted with genomic templates of a positive control, which is Haemophilus influenza, a negative control that can be another species of Haemophilus genera, and a black control at 63 Celsius degrees for 1 hour. Then, to assure these results, the mixtures have to be tested with LFB. The results obtained here can confirm that the primers were suitable for only Haemophilus influenza detection with LAMP-LFB assay. Sensitivity and specificity. Specificity of the LAMP-LFB method has been evaluated with genomic templates extracted from 10 Haemophilus influenza strains: Haemophilus parainfluenza, Haemophilus hemolyticus, Haemophilus parahemolyticus, and non-Haemophilus influenza bacterial pathogens. All results indicated specificity of Haemophilus influenza by LAMP-LFB, which was 100% specificity for Haemophilus influenza detection. The limit of detection of LAMP-LFB assays was 100 femtograms by the 4 detectioning methods.
Some advantages of the LAMP lateral flow biosensor test are that it has a high efficiency amplification of DNA under isothermal conditions, so the reaction is easy to perform in a regular laboratory water bath or heat block. It only needs a few DNA copies to detect the analyte, and its detection is simple, easy, and selective. It also has a high specificity to the target DNA sequence. On the other hand, lateral flow biosensor is more sensitive and accurate than agarose gel electrophoresis, colorimetric indicator, and real-time turbidity, and it is also simpler and faster, and is not expensive. However, there is a limitation for the detection of Haemophilus influenzae by the LAMP lateral flow biosensor, since the LAMP results are shown qualitatively by red strips. Currently, there is no laboratories that sell a LAMP lateral flow biosensor test for Haemophilus influenzae. However, there is an estimated price that ranges from $2 to $6 reported for Candida albicans and for SARS-CoV-2. Thus, we expect that the LAMP lateral flow biosensor test for Haemophilus influenzae costs about $5 to $6 per disposable test.